Good night, wherever you are. We are pleased to welcome Professor Helge Bruelheide to our international seminar series at the graduate program in ecology of the Federal University of Rio Grande do Sul. Professor Bruelheide works at the Martin Luther University, Halle-Wittenberg, and at the additive, which is the German Center for Integrative Biodiversity Research in Germany. Helge, thank you very much for accepting our invitation to present the seminar for us. And the floor is with you. After the seminar, questions will be welcome. So please, you can write your comments, your questions or comments in the chat that you can see through the YouTube by streaming.
Okay, Valerio, thank you very much. Muito bom dia, foi com grande prazer que aceitei o convite do Valerio Pilar para dar uma palestra aqui no seu seminário. I have to excuse that I switched to English now, because otherwise I'm afraid that the content of what I'm talking about would be hardly understandable. So I will speak about the mechanisms driving biodiversity ecosystem functioning, or in short, BEF, in forests. I will do that using our own results of the BEF-China experiment in the subtropics, which is probably the experiment with trees where these mechanisms are best understood. I would now love to share my screen. Is this possible? Excellent. Thank you so much. So I'm doing that on the perspective of one of the founders of the BEF-China experiment, which was established in 2008, and of one of the spokespersons of our Chinese-German graduate school called 3D, which is Chinglish and stands for Land, or forest land in English. And it's run by my university, Martin Luther University, Halle-Wittenberg, and UCAS, the University of Chinese Academy of Sciences. And this explains all the logos you see on the first slide. So after a short general introduction on the objectives of BEF research, I will speak shortly about observational studies. So what we have learned from studies in natural forests, and then I switch to the experiments. And the main part of the talk will be about the 5 mechanisms that might play a role to explain the biodiversity ecosystem function and mechanisms. The species identity, the first, so that species with particular traits might result in particular effects, selection effects. Then niche differentiation, so the divergence in functional traits among the different species in the community, which might result in an increased resource use efficiency or in an increased proportion of the resources captured. Then the third are increasingly positive interactions. So we have less competition, but we have more facilitation. Then we have dilution effects. So negative effects you usually have from herbivory or pathogens decrease because the resource these organisms live on, they are diluted. This is also part of the Janssen-Cornell idea. And finally, we have trophic networks, so that there is a top-down regulation of herbivory, for example, which might play a role. And I will explain all these mechanisms in the second part of the talk. So why are we doing BEF research? The overarching objective is to assess the role of species diversity for ecosystem functioning. And this can be, for example, simply productivity. You see a tree growing here showing increasing production, as a result of increasing biodiversity. Productivity is a typical ecosystem function that is measured everywhere. So it's a property or ecosystem process, you could say, that's in principle neutral for humans. But there are others as well, like, for example, soil development, nutrient cycling, provision of stability, or here, as you've seen, maybe the prevention of soil erosion. Yeah, you have less erosion if you have more tree species in this community. And those functions that have an appraised value for humans are called ecosystem services. So I deal with them together because many of the functions are also services, but not all of them are. More recently, the ecosystem services are also named NCPs, Nature's Contributions to People, but in principle, they mean the same thing. We have learned a lot from observational studies, and as I said before, I will focus on forests. And the most comprehensive paper so far is the one led by Jingjing Liang, published in Science 5 years ago, where they compiled data from 780,000 different plots, forest plots, and demonstrated an overall consistent positive concave down relationship between productivity on the y-axis and species richness on the x-axis. And the strength of this paper is that the authors controlled for the environment. So they put in a function in here that controlled for variables that might also affect this relationship, like the stand basal area, climatic, soil, topographic, covariates, and so on. And they found that this exponent here, they call it theta, the elasticity of substitution, which is the strength of the effect of diversity on the productivity, was significantly positive in a lot of different forest types. The boreal forests in North America, Northeastern Europe, Central Siberia, East Asia, Central Africa, Southeast Asia, Malay Archipelago. So overall, this means globally, a certain percentage of loss in forest tree diversity would also result in a greater percentage of loss in forest productivity. So the authors pointed out that this is a global issue and an economic factor. Productivity is only one of the different ecosystem functions. When we did a large European study in European forests running from Finland, Sweden, Germany, France, Spain, and also Romania, with 210 plots and different forest types you see here on the bottom. We also studied a lot of different functions here. And these are the columns of this graph. It came to functions like timber quality, the amount of timber, but also regeneration, the root biomass, but composition, for example, of wood and of litter, microbial biomass, so life in the soil carbon stock. And then we had a lot of other biodiversity levels, like, for example, pathogens and herbivory. And you can then also express the lack of herbivory, so the complement of herbivory as an ecosystem function. Yeah? The less herbivory, the better the forest is functioning. The fewer pathogens you have or damage you have, the better it is functioning, and so on. So all these columns you see are here in this graph, and on the rows you see the different tree species we had in our European observational study, starting with Abies alba, the fir, and ending with Quercus rubor, the oak. And all the reddish colors here show that this particular species is very good for certain function. Yeah? So for example, Abies alba is very good for timber production, but it's not so good for bat diversity. You know? The bats really don't like these conifers. And for that reason, if you put this together, you see a salt and pepper picture. And you can also see that there is no tree that is good in all aspects. Yeah? And you can also see that these different functions are not fully related to each other. So there's not always red in the same row, for example. Yeah, and because of that, it is possible that we can have different effects of different groups of organisms on the different functions. Okay, what we did is a similar thing using the forests in China. And I will speak about the experimental sites in Jiangxi, which are the green spots here. In the neighbor province, that's Zhejiang Province, we have a natural forest. It was a nature reserve. Now it's a national park, just in the corner of this province. So here's Shanghai for the overview. So this is Zhejiang with Hangzhou as the capital, and here's Jiangxi. And exactly here on this border, we have both our comparative study plots, where we studied 27 plots for different functions, but we also did our experiment. I come back to the experiment in a second, but we stay with these natural forests for a second. And it was Andreas Schult who summarized again different functions in these natural forests, starting with erosion control. We also had microbial activity, we had leaf and wood decomposition, we had also herbivory, predation, and parasitism. So predation and parasitism was measured because we exposed artificial trap nests for insects there, where we could count the insects in these traps. And the pattern is a bit different here, because we don't have species here on the rows, but we have different predictor variables. Yeah, so it's not the particular species, but it's here, for example, saprophytic fungi richness, yeah, which is negatively related, for example, to end cycling. So red dots means a negative significant relationship, and blue dots mean there's a positive relationship. And also here you can see that there are different groups of organisms, like decomposers, that have positive effects on different functions. Yeah? And not all of them is related to each other. But the most important point here is you have also a strong effect of environment. We brought in the environmental variation by using a PCA. So we have the first and the second PCA axis to summarize that. And this means whenever you want to study such a thing, like effects of certain organismic groups on functions, you run into the problem that you have to control for the environment. So what Ling Jiang did with including an environmental function in this relationship, but which is really difficult to do with only 27 plots. Yeah? So you always run into the risk that you might interpret an environmental signal as, on erosion control, for example, as a relationship to another factor. It's also interesting, you see that on the left-hand side, that you have here all the functions summarized. This is the average multifunctionality. So what Andreas did is he made from all these functions, these columns, one single variable simply by averaging it. And you can see that, for example, some predictors have an effect on this overall functioning, irrespectively of their trophic position. For example, you have an effect of parasitoid richness on the overall functioning. This is significantly positive. And when we did this calculation, so parasitism here, this column was taken out of the multifunctionality so that it's not predicting itself. But this means overall, the more parasitoids you have in the other functions, you have increased functioning. And it's not always positive. You have also negative relationships. And for some variables, that was surprising. So woody plant richness, tree richness, we did not even find a significant relationship. Yeah. So woody plant richness, it's shown here. But if you take out things that are related to that, it's no longer significant. And this shows the main problem of these observational studies. Yeah, you always have to control for the environment, and environmental covariates might interfere with your results. And for that reason, we have come to the conclusion that you should study such relationships experimentally. Because if you want to study this, what I have shown before, you have to take out the back loop. Yeah, it might also mean that a good ecosystem functioning, well-performing ecosystem also promotes biodiversity in natural systems. And then with this feedback loop, it gets really difficult to disentangle cause and effects. And this is what already the famous poet Johann Wolfgang von Goethe said: one is inclined to mistake cause and effect. So if we're doing that, we have difficulties to tear it apart, despite all the statistics we have for observational studies. Yeah, and this is why we need experiments. This is my favorite slide. I like to show to my colleagues in molecular sciences, because this is so familiar for them, how they study biological systems. And they do that by removal. Yeah, they do that by removing important components. So if they want to see how certain combination of genes affect a function, they simply start to take out one of these genes. And then you see a loss of function, and you know, okay, this gene was somehow important for the function I would have loved to observe. And the idea is the same for ecosystems. Yeah, you take out one plant species or two, and then you see, okay, there's a loss of functioning. Yeah, there's maybe less decomposition, or there's more herbivory. And actually, you can do that. And there are nice experiments out there, removal experiments, and I know that also Valerio Pilar is running some of them, where you can easily do the same thing and simply create systems where certain components are missing because you have removed them. The problem is you cannot do that easily in a forest. If you do that, you get not a new system with fewer tree species, but you get a forest with gaps. And in particular, if you do it in an old growth forest, and then you have something completely different. You have gap dynamics, you have much different microclimate, and so on. And this means for forest biodiversity experiments, we cannot go this way. And for this reason, we have to go the way to create these systems from scratch. So we create artificial, synthetic communities that do not exist in nature. And this idea has been done a lot of times with grassland experiments. It has been done, of course, also with mesocosms and bacteria and algae and so. But with so far, the most convenient way to do it with vascular plants was to use grasslands. And here you see the Jena experiment a few years ago, where you had a pool of 60 plant species on large plots of 20 by 20 meters. There are also some smaller plots here, these 3 by 3.5 by 3.5 meters, but the large plots are actually was the main experiment. And the question is why we couldn't stick with that. Why did we have to move to trees? Why it is so interesting to switch from grasslands to trees? And one of the answers here is that these BEF relationships, if you look at them closely, are mostly driven by plant density. So this is one mechanism I'm not speaking about because it's so obvious, but it's not well known in many cases. In the Jena experiment, it was shown, for example, here on the left-hand side, you see the positive relationship between productivity above community biomass on a log scale. So this is 1 kilogram here. And it's increasing with increasing species richness, 1 to 16 species. But what is not increasing is the size per plant. So if you really look at every stalk of grass and every herb, you don't see an increase in the plant size. Yeah? So the size in gram per plant is the same. And this means you can only get this relationship if you have a higher density. Yeah? So if you count the number of modules, the number of stalks, grass halms, you could say, on a certain area per square meter, you see that this is increasing. So that means in the high diversity treatments, you simply have more individuals, and these individuals make more biomass. And this is difficult to control for. We also see that in forests. So in the natural forest, I have shown there was one PhD student, Martin Barrowful, who did that. And he also could show that there's a relationship between the increase in basal area. So this is the productivity in forests with tree species richness. And if you look at the response, the delta BA, delta basal area, you can see that most of the effects go through density in natural forests. Yeah? So you have, with the higher diversity, irrespective whether it's species richness, it's phylogenetic or functional diversity, you have an increase in density, and with an increase of density, you have an increase of basal area. So here the red colors are positive relationships. The blue ones are negatives. Again, we have age here, so there's a confounding with other factors, but this is not so important. The main point is here, the main arrows in this structural equation model go through density. And density is something we can control for in experiments. Another effect is that trees have a memory. And this means that these BF effects accumulate with time. While in grasslands experiments, you have more or less the phenomenon that you have this more or less same relationship in every year. Yeah. So this is the relationship for different years. So the different lines here are any year between 2003 and 2007. Yeah. So sometimes it's lower, sometimes it's higher. This depends on the climate. If it's moister, then you have more biomass, but it's more or less the same slope in all the years. This is different for our forests. This is the paper from our group published by Yuan Yuan, the PhD student in the project in Science a few years ago, where you can see for the different years that you have not only an increase in the standing biomass, but you also have an increase. So biomass is the above graph, but you also have an increase in the volume increment. So how much wood is produced every year? It gets more and more, and this relationship gets deeper. Yeah? So we have a positive effect of time on the BF relationship, which is highly interesting and makes it worth studying. Finally, we also have stronger stability effects in trees, and this makes it also interesting. So this is a graph from Florian Schnabel, who is one of our PhD students in our graduate school, and he shows this how this works. You have a normal year where every tree is growing normally. So you have an increase that is average, I would say. And then you have a drought year, yeah, shown by the sun, where only one tree species is really producing a lot. Yeah? So this is this tree here, that's above average. So it can cope with drought. In a wet year, it might be a different tree. So in the wet year, another tree takes over, and then it's this tree that produces more biomass, while the drought-prone tree the drought-resistant tree might produce next to nothing. Yeah, but on average, you still get the mean productivity. And if you calculate it across different years, and Florian did that for the BEF-China experiment, also using the years between 2010 and 2019, over 9 years, and calculate the stability, which is the mean divided by the standard deviation. So it shows how much it's varying between the years. Then you can see that we have a positive relationship to species richness. And this is also a very nice thing that you can do with these long-lived organisms with the trees. So for these reasons, we love trees for the BEF experiments. It's not only us who love it. Meanwhile, a lot of experiments have been established. There are also 2 experiments in Brazil, the Brazil Dry and the Mata Diff experiment. They are more and more in the tropics, but most of these experiments are in the temperate zone. Yeah? So like the ident experiments with a lot of sites also in Europe. The earliest experiment ever was in Satakunta, the Boreal experiment by Julia Korycheva. But we also have now 2 in the Mediterranean, in Australia and on Sardinia. And the only one so far in the subtropics is BEF-China. And BEF-China is actually also the largest one of all these experiments. So we have planted about 500,000 trees and shrubs, and we are contributing, I think, about 40% of all these trees to the whole network of these 27 experiments we have globally. BEF-China is divided on 2 sites, so these are independent replications with independent species pools, each on a site of about 20 hectares, and each of these sites with about 300 plots. Yeah? The plots you can see here as a projection and below these 2 sites, and the different colors show these different species richness. So we have created monocultures in yellow, but also 2, 4, 8, 16, 24 species mixtures. You have in total 566 different forests, varying in species composition and diversity. And because this is an experiment, they have been assigned randomly to all these positions, and this is why we can control for environmental effects. And this was the first tree ever planted. It's funny that this is a monoculture of conifer, so it's a commercial monoculture of Pinus masoniana, but that was, yeah, just chance. And I had the honor to do that. I had some help, so we had hundreds of people helping us to get the whole thing planted. And it looked like that in the beginning. So 1 year after, no, it was half a year after planting in 2009, you could still see where we put the plots. So each plot has an area of 666 square meters, which is 1 unit of Chinese area, 1 mu. And so here we put the plots. They look very bare because you can barely see the seedlings, but 8 years later, you can now pretty well see that this is a monoculture, H14, for example, and this is a mixture, J14, and so on. But this is only a very small section of the whole experiment. As I said, these are just a dozen of plots you can see here, but we have 566 in total. So what came out of these experiments? I told you we have these different mechanisms, and one of the most important mechanisms is are species identity effects. The different species simply don't grow equally well. We have very, very well-growing species, for example. So the best candidates here are Nyssa sinensis, Correspondius axillaris. You also have Triadica cotuchinensis, which was Sapium before. We also have very poorly growing. So the Quercus filioreidus grows poorly, and also Meliosma did not grow well. Yeah, so it depends on the species. There are clear species identity effects, which we already have seen in the first years. We can also relate these effects to traits. Yeah, so this shows the overall trait composition of all these species. The species in black letters, the traits we have measured in red. And these are different categories. We have crown traits like the crown length or the crown size, the crown ratio, the ratio of crown diameter to stem diameter at ground height. We have physiological traits. We measured stomata conductance. We have water potential and the mass-based contents of all the elements. And we have morphological traits, so leaf area, but also specific leaf area, leaf toughness. And we also measured morphological characteristics like the thickness of palisade parenchymes. So if we run the model, the best model consisting of these traits, it's really disappointing. So we could only explain about 42% of all the variation using all the traits. And these traits only played a role in combination. So there was not a single trait that explained best the production. Yeah. So of course SLA was included, but it's not. It was not that strong as we had expected from the leaf economic spectrum, for example. And these were different traits related to the structure, but also traits related to water use, but also traits related to photosynthesis. Yeah? So it's a bit difficult to explain why certain species have this strong species identity effect. More recently, just published, we analyzed these data, and we have as rows all our species here, and we could estimate the effect of species identity for all these species. The interesting point here is that all significant results we have here. So these are the points with the black point and with an error bar that is not overlapping with 0. All these are positive. So we only see positive selection effects here of the. Oh, sorry, it's not a selection effect. It's a species identity effect on these BEF relationship. And I think about 17 of these 40 species had such a significant effect. And again, the most productive ones are Liquidamba, Nyssa, Corresponias, Alnifilum, and also Shima supera. Again, it was not easy to relate that to traits, but I have said that before. What the group found out is that the typical traits we had assumed were not significant. For example, this is the percentage of evergreen species. We would assume that these evergreen oaks we have there, they grow less, and that evergreenness, so the leaf habit or leaf longevity, you could say, had a negative effect. There is a slight tendency, but it's not significant. Yeah, it's only an 0.067 significance. However, what played a role was the percentage of species with an avuscular vascular mycorrhiza. And this makes it highly interesting. We know this globally, that the AM trees, of which I have also a lot of in Brazil, for example, they grow better than the ectomycorrhizal trees globally. And this is also the case in our experiment. It's still not clear for me whether this is a causal relationship or whether it's a side effect. Yeah? But we could, of course, assume that this has an effect. So AM species might take up additional nutrients and might be more resistant to abiotic stresses because they have this soil-dwelling avuscular fungi. Or the other way around, the EM species, the ectomycorrhizal species, they might have a disadvantage because they are more susceptible to soil-borne pathogen accumulation. We know that for EM trees, for ectomycorrhizal trees, for example. Yeah, but it's still open to explain this pattern. So next effect after species identity is how you combine these different trees. Yeah, and this is the realm of niche differentiation. And this can be best explained by a graph that had been published by Laura Williams from Janine Kavanagh-Bear's group, found in one of these ident experiments. So what they did is they calculated crown space filling. Whoops. My graph is gone. Just a second. I have no idea what happened here. Okay, my graph is gone. Doesn't matter. Space filling is best explained by crown overlap. You have two trees growing together, and then you calculate the amount of overlap between the two crowns. Yeah, and the amount of overlap is called the crown complementarity index, CCI. And they could show that CCI increases with species richness. And they did it manually. So they measured everything with the measure stick, which was a bit tedious and it's not very accurate. What we did is we simply repeated that in the BEF-China experiment, but we used terrestrial laser scanning. So a terrestrial laser scanner takes the whole three-dimensional picture of the whole stand, and then we can calculate the overlap in crowns. And this was done by Matthias Kunz and his colleagues. So this shows how that looks like. You transform all the points that are hit by a laser into so-called voxels. So these are three-dimensional pixels in the picture, and you can calculate how much of these voxels occupied by more than one individual. So this is seen here, which is the crown overlap. And from this, you can do the same calculation as what can be done manually. This is the crown complementarity index here. And you can see that this overlap is increasing with increasing richness of the neighborhood. So in principle, we could have 8 different neighbors, but not all the positions were filled in this experiment. So some trees died. That means the more neighbors you have around, no, the more different species you have surrounding an individual, the more crown overlap you have. Yeah? And this has explained a lot of our over-yielding or net biodiversity effects. Another way to assess the niche differentiation is simply to look at the functional traits. And I know it's not that easy and simple, but let's assume for a moment that we can equate all these trait identity effects with community-weighted means. Yeah? So we simply calculate what is the mean specific leaf area across all the different trees in a plot. And as you can see, the community-weighted means are here in blue. This effect on productivity, this is the slope in a standardized regression, is increasing. Yeah? The longer we had this experiment running, the more important the mean of all these different traits we have been analyzed became. Yeah? So trait identity is becoming more and more important. But what is more important is that we also have a measure of niche differentiation by simply looking at how different traits complement each other. So you have a high functional diversity if you have, for example, a tree growing together with a high and a low specific leaf area. And this is again summarized here across all the different traits. In the beginning, functional diversity was less important than community-weighted means. But after 8 years, it became more important. And now it's far off the community-weighted means. That means with time, these niche differentiation effects outweighed the species identity effects. And this is also shown by the different categories. We have community-weighted means for different traits, hydraulics, for example, like water potential or hydraulic conductance. But we also have that for chemical traits like nitrogen content, for example. And you can see that in particular the hydraulics, they play a role in red, they play a role in the community-weighted means, but also in the functional diversity. I have removed my laser pointer, I see. Now it's better, I think. Yeah. So this is the reddish graph here. You can see the functional diversity effect is steadily increasing, in particular of hydraulics. That means combining species that are water savers with species that are more conservative in water use is one of the main reasons we have a positive biodiversity effect on productivity. Yeah? So this is also a very important result for devising new forest types in the future. The third mechanism I would like to speak about are positive interactions. And this was an idea by Andreas Fichtner of Goddard, of Werner Hertle's group, and Goddard von Oheim was involved here. So we always try to plot this competition intensity against neighborhood richness. We thought there is an alleviation of competition if you have more species in the neighborhood, but it really didn't work out. And then Andreas came to the brilliant idea to do this separately for the acquisitive and the conservative species. Yeah. So what you see here is a PCA of all the traits. We have 4 traits shown. This is the leaf economic spectrum. On the left-hand side, you have the high SLA acquisitive tree species, mostly deciduous ones. And here on the right-hand side, you have those with higher leaf toughness, and these are the more conservative ones with the thicker leaves. And if you plot them separately, it nicely plays out. So for the acquisitive ones, you have a relative interaction index here, which is calculated by growing with neighbor compared to growing without neighbor. so growing alone. And this is possible because we have this situation because there are some trees without any neighbors. They, of course, grow better because they have more space and they don't have competitors. Yeah? And for the acquisitive species, they're all negative. All these lines are negative, independently of the tree size, which is shown here. And it gets more negative the more species you have in the neighborhood. Yeah? So competition gets more severe with more neighbors for the acquisitive species. For the conservative species, it's just the opposite. The conservative species, they are also negative in the beginning, but then they start, after having more than 3 neighbors in the neighborhood, different neighbor tree species, they have a positive effect. And positive effect always means facilitation. So these trees, the conservative ones, they grow better with more tree neighbors in the neighborhood. In other words, they don't suffer from competition. They are favored by neighbors, and this is called facilitation. And it's one of the rare cases where you have facilitation, which is not brought about by legumes and nitrogen fixation, but really it's brought about by normal neighbors. So Andreas produced a model out of that, the neighborhood model. It's the neighborhood model you can see here. And we applied it from site A to site B. It was just the model used and then projected on the other side, completely independent replication. And what we have seen is that this explains pretty well the above ground productivity. And this graph here shows the contribution of these different factors. It was a mixed model, so we have fixed factors and random factors. But this fixed factor, this model, neighborhood model, explained in more than 50% of all the explained variants. And overall, we could explain the marginal R square, about 60% of all our tree growth by our fixed factors, or even if we included the different communities we had as a random term, we could also explain 80%. And this is extraordinary. If you ever have tried to predict tree growth from traits, as we did before, you reach maybe half of that. But showing that the neighborhood of a tree plays such an important role, and simply by using acquisitive and conservative traits as a concept was really very, very amazing. Then our fourth mechanisms are dilution effects, and this can be best illustrated by pathogenic fungi on the leaves. So over all these years, we have identified about morphologically about 100 different leaf fungi. Some of them were new to science. We have also described new species that were so far unknown, but this is not what I'm talking about. We could do that because we teamed up. So Mariam Zadani is the PhD student in this project. We had people experienced in morphological identification, Uwe Braun here, and we had a couple of people working in the field, like Lydia did that, and also Steffen Bien did all the analysis morphologically. And finally, we also analyzed the OTUs by extracting DNA and doing metagenomics with these leaves with the help of Tess Freiburg. Yeah, so we put this all together, and the result you see here. This is the mean fungal infestation. It shows how much of the surface in percent is covered by these fungal pathogens. Yeah, you simply count these black dots, and then you have the percent of leaf damage. It's on average 10%, so it's really high in the subtropics, but it's decreasing the more species you have in the neighborhood, expressed by Shannon index. It's not that clear for the OTU richness, yeah, because it's simply dependent on the different species. So it's significant, for example, for Triadica zebifera, Sapium zebifera, but it's insignificant, for example, for Sapium Sapinus saponaria. Yeah, so it's more difficult, and we are still not sure whether this is, might be also the effect of contamination, because these samples were not surface sterilized, but actually more or less we have the same pattern. The interesting part is it's all explained by dilution, because if we change the x-axis from Shannon diversity to the proportion of target species, that means the conspecific species, Yeah? That means here we have all the monocultures, and here we have completely different species in the mixture. You have a fewer infestation rate. It's not significant for the OTU richness, but for the infestation, it's clearly significant. Yeah? That means the amount of pathogens you have on a tree depends on how many other tree species of the same tree species you have in the surrounding. Or the other way around, if you dilute a certain species, it can hide among other species and is less affected by the pathogens. And of course, this has a strong effect on ecosystem functioning. We thought we would see the same for herbivory, but it didn't work out. Herbivory is positively related. And this is a story I can maybe speak about more later when there are questions. So we have a higher leaf damage the more tree species we have. And I skip these two slides because it's a bit short in time. Both of them have effects on relative growth rates. So again, we have the productivity here shown not on the y-axis. We have it here on in the color scale. So we have a huge production here with about 0.7 relative growth rate in dark red in the top corner here. And we have more yellowish colors on the left-hand side. And that's really amazing because what we see here is that we have an interaction of herbivore damage, which is just the opposite. Yeah, so the herbivory damage is here highest, where we have the highest production. So we see something like an overcompensation. The more the leaves are eaten, the more they are producing also wood. That's really amazing. For the herbivory, it's a very strange pattern. For the fungi, it's also very funny because we here see with increasing pathogen damage, the more fungal infestation we have on the leaf, we have a lower production. So the yellowish colors are here. But this is only relevant at high species richness. So this relationship between damage and the fungal load only plays out at high species richness. In monocultures, it really doesn't play a role. Yeah, and these are very difficult results to be interpreted, and we are still struggling to put it together. But it shows that these different mechanisms, they're not just going all into the same direction, they might also outbalance each other, which makes it even difficult in experiments to disentangle them. And finally, I have to come to an end now. We have the trophic networks. The idea is that with presence of trophic networks, we have a top-down control on the producers in the end. And the idea was here to study the ant aphid mechanism. Yeah, so we have aphids sucking on the leaf, yeah, doing damage on the leaf, but we have ants tending the aphids, and the ants also take away some of the caterpillars, so they're also predators. And the idea was that we have less herbivory damage when we have this trophobiosis between ants and aphids present. But it wasn't the case. Overall, we had even a positive effect. Yeah, so we had more leaf damage when we had ants and aphids. But again, the story is more complex, yeah, because this damage was only increasing with the number of trophobioses here in the monocultures. In the high species richness plots, we had this is expected negative decrease. So we even have, for such effects of these trophic controls, we have interactions with the number of different tree species involved. The next steps we are doing is we construct networks, which we did here for the aphid ant networks. What you see here are the different levels. So we have here the plants, 16 or 15 different species. Then we have the aphids here. So this is one network, which aphid species is sucking on which plant. And then we also have the ants tending on the aphids. And this is the monoculture, and this is the 16 species mixture. What you can see is these networks get more connected, or we could also say they get more general. The generality of these networks gets higher, and generality is a measure of how many species of the higher trophic level are connected to the lower trophic level. This seems to be also the case for the soil fungi, but actually we don't have full conclusive results here. We are still struggling with interpreting that. This is a recent paper from Christine Weisbecker, who did that for the soil fungi in all the experiments. Again, it makes the impression that generality is higher at higher species richness, but this is not yet fully established. So let me come to the conclusion of my talk. I have shown that these 5 mechanisms all play a role. We have these species or trait identity effects, which are clearly established. We start to understand them, but it's not that easy that there's only one trait. We have the niche differentiation, which is developing with time and has a very, very strong effect. And maybe the best established result we have is for the increase of positive interactions with increasing local neighborhood richness. We have for the dilution effect so far only inconclusive results. So it's positive for the leaf fungal pathogens, it's negative for the herbivores. And for the trophic networks, actually we don't know yet. Yeah, this is still to be established. We are working on that. And this is also what we will do in the next steps. We have the idea to now look at the partial explanation of all these different mechanisms on the overall ecosystem functioning. Yes, I think we now have the data together that we can do that. We can simply say, so how much is it niche differentiation? How much is it a dilution effect? Yeah? But again, there are limits of how far you can go there because it's the same experiment. Yeah? And we have not disentangled these effects. So for that reason, we, for example, would have to create experiments with different densities, again, to show that we have a dilution effect. We're also focusing on the interactions between the producers, in this case, the trees and shrubs. This is the International Graduate School 3D. And we are also planning to do a new research unit with a zoological focus, where we ask how much these across trophic level relationships, the networks, play a role. So that will be the future of the next steps. I hope you enjoyed this short introduction into all these effects. I have to thank a lot of people, and the list is far from being complete. So many people have contributed to BEF-China over the years. More than 100 researchers have done their work there. I also have to thank my funding organizations. In the forefront, it's the German Research Foundation, DFG, who funded first our research unit and then now our graduate school. And I'm also very happy for funding I received through additive, through the German Center for Integrative Biodiversity Research. I thank you very much for listening, and I'm open to questions.
Thank you, Helge. Fantastic to hear your presentation, to see your presentation about these BEF experiments. And we may have questions in a few minutes. I do have a question regarding the experiment in China. How did you choose the mixtures? So how did you choose which species would be put together? So did you have all the combinations, or so how did you select the species and the mixtures?
I mean, there is a technical answer to that, and there is an honest personal answer to that. So the technical aspect is we used a broken stick scenario, which did not use all the species combinations. This is impossible. That would be more than 1 billion of combinations. You can combine 40 tree species. So we made sure that we started with 16 species, and then we broke it down in 8-8. Yeah? And these 8-8 mixtures were then broken down in 4 times 4, 8 times 2, and 16 times monocultures. That made sure that every species occurred only once at every richness level. And of course, we missed certain combinations. Yeah, that's perfectly true, but this is the way it is. Species selection, this is the personal answer. We were not sure what we should do. So in the beginning, we were asking all the experts in forest science, hey, which species should we use? And they said, oh, forget it. This cannot be grown alone. Nobody has tried it. It's not a commercial species. So what we did is we raised more than 100 different species, and then we used those of which we had enough seedlings. No? It's just chance, I would say. And I'm very happy that this played out so well. I think we have about, I don't remember, I think we have 21 deciduous and 19 evergreen species nicely mixed. We have ectomycorrhizal and arbuscular species randomly mixed. So it's, I think in the thinking about that later, it was a very good selection, but we were simply lucky because these were those species of which we had enough seedlings. We needed more than 8,000 saplings for every species in the main experiment. Yeah. And if we had only 2,000, we couldn't use it. So we had huge nurseries. We raised more than 1 million trees, but we could only use 1/4 of that.
Thank you. Thank you. We have a few more questions. Fernanda Teixeira is asking, thank you the presentation, and please can you talk more about the relationship between higher leaf damage and higher number of tree species?
The leaf damage. Yeah. There are several aspects involved. So I think the one important aspect is the transmission. If you have susceptible tree species hidden among neighbors that are not competent for this particular leaf fungi, you simply can hide between other trees. Because those trees don't get infected, you have a lower chance that the spores can cross this larger distance. Yeah, this means what you're doing with the dilution is you simply increase distance between the source and the target tree. Yeah? I think this is one of the most important mechanisms, but it would be clever to demonstrate that. I think nobody has shown that yet in one of the experiments. But there are also other explanations. We also see microclimate effects. We simply have, in the mixtures, we simply have a more balanced microclimate. So we have less temperature extremes and we have a higher moisture in these stands. And this is not favorable for a lot of our fungal pathogens. Yeah? So it might be that there is also simply a microclimate effect. You can test for that because the microclimate effect is not dependent on the identity of the neighbors. And we did that by looking at all the different identity effects of the different tree species on the pathogen load of the target tree. Yeah, so of the identity effect of non-target tree species in the neighborhood on the target tree pathogen load. And we did that first, so Lydia Hönig did that first in the German Kreinitz experiment, and there are a lot of these non-target effects. So it's not only the dilution by the conspecifics, it's also some other factors like microclimate which we do not fully understand.
Thank you, Helge. Another question is from Antalya Quinteiros. I wonder if you could explain the difference between trait identity effect and the species identity effect. Are they the same?
Yeah, that's a very good question, and I apologize if I also tend to mix it up. So what we can clearly establish are species identity effects, yeah? Simply by looking at the particular role a particular species plays in this experiment. A trait identity effect would be that we have a certain trait state that can fully explain this species identity effect, and which we really cannot see. So there is not a single trait that would explain these species identity effects. So what we have on the other side is a combination of traits, and I still think we lack important traits. Yeah, so we don't have the root traits. We don't have molecular traits. Yeah, so there's so much. There's so important traits on the molecular level. The amount of phenolics, the amount of alkaloids, for example, whatever. So I think we missed that. For that reason, I would not speak of trait identity effects. So sorry for doing that in the last slide. Actually, what is sure so far is that we have strong species identity effects, but so far the trait effects are quite heterogeneous, I would say. So I would make a difference here.
Thank you. Another question by our colleague, Sandra Miller. She's interested in the positive interactions. Yes. Did the differences in the growth rate of acquisitive and conservative species consider only the total neighbors' richness or also the trait composition or maybe diversity of such neighbors?
Only the total neighbor's richness or the trait composition? Good question. No, we did not take into account the trait composition. This was only against the neighborhood richness. But you are completely right. Would be interesting to do that. I think it's a brilliant idea to do that. And this is something we have started to analyze in the international graduate school, because here the idea is that we replace these richness effects by trait effects. So a richness effect would be the Shannon diversity of the neighborhood or the number of neighbor species in the neighborhood. Yeah? And if we could replace that by, let's say, a functional diversity effect of relevant traits, we would be more general. Yeah? It would provide a more general explanation for this effect. I think this is a brilliant idea to go forward and maybe to try it out in one of the next analysis. So thank you for this suggestion.
Well, thank you, Helge. I see there is, many people thank you for the seminar. And our colleague, Milton Mendonça, is asking, in the experiments, plants are introduced purposefully. Are other species in different trophic levels also introduced in some way? So the plants were controlled, but the other trophic levels were not. Do they come from neighboring areas?
Yes, also a very good question. I mean, this is the type of experiment we should do in the next generation. Yeah? So we have to manipulate the other trophic levels and keep the producers constant. But this is not that easy, you can imagine, because herbivores move across the sites very quickly. The same is true for pathogens and other species. So no, all these other trophic levels came in from the surrounding, and this is also a big advantage of the BEF-China experiment. It had been established in a forested landscape. So Jiangxi Province is the province with the highest forest cover in China. They also have a high amount of natural forest, and we used a plantation of pine, of Pinus masoniana. So it was surrounded also by mixed forest, and that means that I assume most of these higher trophic levels have been present and could easily invade from the surrounding. Yeah, so this is what happens. It's extremely difficult to do such manipulation of these experiments. We did that in the third phase, I think, where we applied fungicide and herbicide to control for certain groups of higher trophic levels. It's simply not possible. We have a precipitation of around 1,600 millimeters. It's raining like hell in summer, and whatever you bring out there, so you using insecticide for the herbivores, it's washed away in 2 days. Yeah, so that means it's easily recolonized again and again. So this is easier to look for such effects in controlled environments. So we are using at the moment ecotrons in Germany for that. So these are mesocosms which we can control for everything: moisture, irradiation and so on, and where we can put in the herbivores or the predators, or we do it without. And then we can see how the whole thing develops. But this does not work with trees, at least not with old trees, only with saplings. It's highly interesting, but this is the way to go for these trophic interactions in the experimental way. However, we are dealing with old growth forests. Our expectation is that we can also analyze these networks and simply look for the network structure to see whether this has an additional effect on the overall functioning, as I said before. But this is, then are the next steps. So we are just planning to establish all the networks for all the different trophic levels. And then we see whether the network components, the network descriptors like connectivity and generality and so on, whether they can explain ecosystem functioning. But again, it was a very good question. Thank you.
Thank you, Helge. We had a comment by Sandra Miller thanking you, and that she and her group are trying to understand these links between neighborhood and the growth of individual species. Yes, but with observational studies. Another comment by Anna Carnaval. Truly impressive work. Thanks for sharing. And we have a question from Daniela Hoss, and I would have this question as well. So, how much of invasive removal you had to have in these plots?
Yeah, Daniela, that's a good question too. So there have been some projects dealing with the non-native species. The idea was also to test for phylogenetic relatedness to alien species. And so we also deliberately planted, in some subplots, we planted invasive species, but it did not go well. So they did not survive well. And this is one of the results you see in many forest experiments. So the forests are not that much amenable to alien species invasions. It's just a bit too shady for them. So far I can see we don't have any strong invaders of alien species. But I think your question was not related to the alien ones, but to the native ones. And I can tell you, in every BF experiment, the most difficult thing is to keep the monocultures. You always have incoming trees. They are simply dispersed by birds, and their seeds come in and then grow up. And if you don't take care, they are simply then turning into multi-species mixtures. So what we have to do is we have to remove these incoming trees, and that was a lot of work in the beginning. So we simply did weeding. And in the beginning, we also had to weed away typical herbaceous plant species. There's one plant species that is invasive in many, many other countries. It's called miscanthus. We have different miscanthus grass species there, and they're really so fast growing and might grow over the small saplings. Now with the closed canopy, it's no longer an issue, but we had to do a lot of weeding. That's a good thing with the forest experiments. So in contrast to the grass experiments, you don't have to do the removal of the non-target species forever, because it stops after 5, 6, 7 years, and then you have a closed canopy, and then it's no longer an issue. But in the beginning, you have to do that. And I mean, this is the answer to that. We had to do that. Otherwise, we would not have kept the design.
Thank you, Helge. We don't have any question listed yet, but I have an additional question regarding the mixtures, whether they are actually occurring in the native forests in the region, So how artificial are these mixtures? So, of course, for a controlled experiment, it makes sense. But for transferring these findings to natural forests, it's another issue. So how natural are these mixtures?
Yeah, it's also a very good question, Valerio. In principle, all these mixtures are possible because we took the seeds from all these species from the nearby nature reserve, where we find all these species growing in communities together in natural forests. Yeah? So in principle, they are all from the same species pool. However, you don't observe two species mixtures in nature. So in the natural forest, we have on a similar area, we have more than 100 tree and shrub species. Yeah, and our largest species diversity was 24 in the experiment. That means we have missed a lot of natural diversity. So these simple species compositions do not exist in nature. And we also see now, after a few years, that we have lost some species. Some species were simply not drought resistant enough. Yeah? So we have lost, for example, in most of the plots, we have lost Ailanthus altissima. It's one of the invasive species in Germany, and I have no idea why this species did not grow well in its native region in China. Yeah? So it's simply not doing well with drought spells of 2 or 3 weeks or so. We also have the problem that when the few evergreen species, when they close the canopy, they're so dark that nothing grows below. And we have one fast-growing species like Shima superba outcompeting all the light-demanding species below. Yeah? So that turns into monocultures in the end. So we have these typical community assembly effects that now kick in after a few years. And this will maybe answer your question. Let's wait a few more years, and I mean, these completely artificial mixtures that really don't work in nature, they will be gone. Yeah? But you're completely right. This is one of the problems. If you design such a thing from the bottom up with a broken stick, you have monocultures, and you have all the other combinations, you come up with funny mixtures that really do not, I mean, they occur in nature, but they don't grow well. Yeah? They have disadvantages. However, I do not know how to solve the problem, because you need the monocultures to calculate the typical complementarity and selection effects. Yeah, it's always compared to the expectation from monocultures. And also, if you do observational studies, you use this information of the species poor plots to inform the species rich plots, yeah? To inform the expectation. And this is one of the rules of all these BEF experiments. You need monocultures. And I would also say in forests, you also need the trees growing alone, yeah, to be able to assess the facilitation and competition effects. You have to do that experimentally, but it's a good question how to make sure that this is not artificial. I fully agree with you. It's one of the difficulties in such experiments.
And also the complexity of such a huge experiment. That photograph with that crowd planting, gathered to plant the trees, is something amazing. Yes.
Yeah,
So
let me tell you, we only employed the first 150 people only in the first week. After that, we found out it's a mess. Yeah, so we organized them in teams of 15, and I think we employed 30 or so in the end, which made it much more efficient. And we never did that again in Site B. So in Site B, it was much, much better organized. Yeah, what you need are teams of well-organized workers who know exactly what they are doing, and then you can plant such a site of 20 hectares within 4 weeks. But it's that was one of the fun pictures in the beginning. Yeah? We would never do it again.
Well, Helge, thank you very much. I don't know if are there more questions. We don't have any more questions in the comments, and we can.
Maybe,
yes, please.
Valerio, maybe let me invite your students to visit BEF-China one day. So whenever travel will be possible again, I would love to also host students and to show you the experiment, also to invite working there. It's a wonderful platform that is, now fully established, and I think wonderful questions can be asked on this platform in so many aspects. And I'm quite sure there are more questions to be asked than we have people to answer them. So whoever is willing to work on that, please contact me. I'm happy to help you doing that.
And maybe we could have a replicate of another subtropical forest experiment around here. Yes. So
I would love that.
Yeah. Okay. Thank you. Thank you. Thank you for everybody who attended. These we had at one point more than 50 people attending. So thank you very much. And this seminar, this is a novelty in our program linked to the pandemic. So we decided to have these international seminars, which are held monthly. So you, Helge, were the third presenter. So it's a way to compensate the lack of possibilities that we are having during the pandemics. So we are somehow meeting here to discuss these interesting topics. Well, thank you. And the live will be disconnected now, and but you can stay, Helge. Thank you.
Thanks again to all the participants, and have a nice remaining day.